Hello and welcome everyone. It was last time in March, and now we're back as promised. I'm here joined with Michael Robin Witt from Gabatter, as we could imagine. And today we're gonna have a topic of EEG and fMRI. Did you have any reactions from the first episode that we were having, Michael?
Yeah, thanks a lot, Marcus. Yeah, absolutely. It was positive, and actually, the nicest thing of the first slide here is that it says episode 2, because after episode 1, people were saying, okay, this is a nice initiative, but make sure that you also come with episode 2 and 3. So, as you said, we're following up. So this is episode 2, and actually some interesting news on the update on everybody's interest in operations on the fMRI data that we got in. And I've tried to make a couple of slides together with Marcus about combining the EEG and fMRI data where we stand now. And then for the next episode, number 3, then we go over to the clinical trial in patients. But now, today is the final summary of the study in healthy volunteers and what we've done with GT002 looking at neuroimaging.
Cool. Yeah. I also had the positive reaction from last time and plenty of questions. I'm also very happy that people followed up and asked questions. And as we promised last time, I think we'll end with the questions. And we'll start off with the slides that Robin and I have been preparing a bit ahead of time.
Yeah, absolutely. But people also like it is very interactive. So Marcus asking question, me answering or not being able to answer. So this time is a little bit at the beginning. I prepared a couple of slides because I wanted to present the data that we got and put the data in context. But Marcus is going to come in and ask slides that are relevant to that particular, questions to that particular slide that we're talking about, and then maybe some more general questions at the end. I think that's the best way to move forward.
Shall we dive in then, Robin?
Yeah, both fees first, as usual.
Yes. So let's start with the first one. You
Yeah,
want to
so
talk about neuroimaging in drug discovery generally, right?
exactly. So, one of the big ideas and vision in our strategy, because this is after all Gabbard insight and strategy, is that strategy is to use neuroimaging methods for drug discovery in particular. And why would you use neuroimaging? Where can you use it? I try to put on the first slide. You can use it for actually from anyway, and how your drug works, mechanism elucidation, where your drug works, how to optimize if you have a whole series, and into clinical testing. So we have actually been using it in a clinical testing in healthy volunteers that we're doing, but we are also now going back on the top of the slide and looking, once we know where the mechanism and how the mechanism might be working on a rather complex scale, like imaging of activity in whole brain regions, then we can use it for further clinical testing. And in our case, validating GT002's effect. Yeah.
I think we've had quite fruitful discussions before, you and me, with comparing, this way of discovering and looking at the drug candidate itself. And I think we're going to come into it here because something that we've been talking about is neuroimaging versus other biomarkers, right?
Right,
And there is some
exactly. advantages
and disadvantages to both of them. Would you like to run it through for us?
Definitively. I mean, of course, everything has two sides. So neuroimaging is but this is also very much, must think of my background being a preclinical scientist in my sort of previous life. I'm always impressed by what neuroimaging actually does. And I've always, forward this idea that neuroimaging is really, for me at least, a window into the brain and the brain activity. And that is very true compared to any other biomarkers. And that is not to say that they are less important or irrelevant or outdated, not at all. But neuroimaging actually does an advantage is at the very first point. You have this a direct visualization of what, in our case, the drug does on the brain structure. That is, I think, already 90% of the ticket for me. And it's also gives you functional information. What does it do while it's functioning? And it's non-invasive. You're not poking anybody. You're not giving anybody any chemicals to do it. It's quantitative and it's visual. I think that's very intuitive. Disadvantage, I think the biggest one is, of course, it's expensive and it's a limited accessibility. Not everybody has neuroimaging techniques, around the corner. And you can do, of course, It's expensive because you can do it in a very cheap way, but the information that you get is not very good. So we actually chose to work a little bit in the hard way to doing both fMRI and EEG, two different techniques. I'm going to go into that later, at the same time in healthy volunteers as a last step before we go into the patients, which is what we're doing now. Other biomarkers, they have the total legitimacy. It could be, a protein that you measure in the blood. It's very cost effective. You just take a blood sample. It has molecular specificity. You're looking for a specific protein, a specific antibody. You can do it very quickly. You have high throughput, not relevant for us. We don't have so many people in our clinical trial. Disadvantages, the biggest one is, of course, indirect. You're looking at that effect, but how exactly is it related to the effect of your drug in the brain? And I think that is the biggest handicap, I think. And if you have the possibility, somebody gives the possibility, do you want to do a neuroimaging? Do you want to do another biomarker in a clinical trial? And I have a chance and the funds to do neuroimaging, I would always go for neuroimaging. And that was basically our idea when we started this project a couple of years ago.
Yeah, because if you look at studies generally, also if you just look at statistical significance, right, it's very hard, get the gut feeling for what's actually happening in the brain if you just look at some biomarkers. But now, what you're pointing out, you actually have an image of how the brain is interacting. That's also very interesting.
Exactly. Let's say you see a parameter that is changed in some project, and then you always get to the question, is this at the beginning or the end of a cascade of a process? But in neuroimaging, you literally see, okay, this is what happens in the brain, in that brain area at that time. After I've given the drug, you can compare quite easily. We did it in the same experiment, a reference compound. You can compare your drug that you're trying to imagine, and that way, have a very direct back-to-back comparison. Having said that, the neuroimaging, the data that you get are very complex. Now you're looking at not at a protein concentration in the blood, but you're actually looking at functional connectivity between different brain areas with different frequencies and so on. The complexity becomes very, very much bigger, and then the interpretation becomes, of course, trickier. But it's the quality of the information, I think. And since we didn't have, 200, 300 patient study, but only 17 and the ones that are going to present, I think that was the way forward. Yeah.
Yeah. Could we dwell a bit more on this, why it's so important with the disadvantage of other biomarkers that it is indirect and there's some variability to it. We gather a lot of data in the way that you're doing it right now. It becomes very specific, but could we talk a bit about the disadvantages with other biomarkers as well?
Well, I think the variability is, of course, something that you have to contend with also in all other methods. As soon as you're doing at the individual measure, you will have large amount of variability, and the variability can be tremendous, can be even in animal models with other biomarkers. Look at it, is definitively a thing. The other thing is, for example, also not only variability, Marcus, but also invasiveness. People ask you, well, have you measured the concentration of your candidate drug in the CSF, in the cerebrospinal fluid? For that, you need to be very, very invasive. It's not nice to do this to people. If I can avoid it, I will do it. And think about it. I mean, at the neuroimaging and some of the data that we've generated, the people were inside the tube, and while they were inside the fMRI scanner, they were doing, a cognitive test and emotional tests, and we were recording them with EEG and fMRI at the same time. So that's absolutely, in my opinion, absolutely brilliant way of going ahead.
So if you look at the left image, although we have separated them, if I understood it right, what you do is you have this helmet of electrodes with the EEG, and then you go into the scan at the same time. Is that correct?
Absolutely. So exactly, Marcus and Jenny already can pinpoint out that in the picture on the left, the woman that is lying on the scan does not have an EEG cap on, which is not correct. In real life, in our experiment, this person that going into the fMRI scanner had the EEG cap on, and they were registering EEG and fMRI signals at the same time. So you can imagine, I mean, you don't have to be an electrical engineer to think if you put so much electrical equipment and measure both magnetic and and EEG and electrical signal at the same time, that you have a lot of noise and a tremendous amount of data cleaning that you have to do, that you're not actually, collecting artifacts, because at the end of the day, what you're trying to do is you're trying to correlate an electrical effect in the brain with a certain pathology or certain behavior to a certain effect of your drug. So it's quite a huge amount of data analysis, and that's actually done by experts. But yeah, picture on the left is correct. Both EEG and fMRI at the same time.
Yes.
And
And then
on
on the
the
right
right, it's
side,
just,
what's
yeah,
yeah,
that's just to
go ahead.
highlight that, EEG is very fast, but it says, you can see this fellow has the electrodes on the top of his head on the skull. It doesn't go too deep into the brain. fMRI is a scanning, which goes slow, but gives you much nicer spatial resolution. Where in the brain exactly is this happening? But at it's a slower level. So that's what these slides.
Yeah, it's interesting also that you would that we've had also in conversation before looking at the time frame. In EEG it's so short, but then you can look over a bigger time span in the fMRI and then see how the brain activity goes over time in a totally different way.
Exactly, so then in that sense, I always said that in a way they are complementary. One is fast but not so precise with the area. The other one is very precise with the area but slower. So in that sense, it makes sense that they're combined. But there, of course, if you have a slow acquisition, a slow getting into of your data, you will miss certain data that's just too fast. You just don't see it. And the other data might get drowned if you take it over too long time. So the two are complementary, and I think it's there are not that many sites actually, when you talk about availability in the previous slide, that you can do both of this EEG and fMRI, and nothing you can just walk in and do this long planning for doing this. But of course now, and there's a next slide, you said, how do you acquire the EEG data? The principle is very similar. I mean, the EEG and the next slide with the fMRI. So you go from acquisition, you take your participants, and all the data today, I'm only talking about healthy volunteers. Always keep that in the back of your head. And what we did was then you have the healthy volunteers. You start with the same person. You make a baseline measurement, for example, in the EEG, you would put on the cap. Or in the fMRI, you put them into the scanner. You do measurements after 2 hours and 4 hours. You do 3 different situations. The resting state, again, this data today is only the resting state. The memory task and the emotion task that we're going to put these people through, we'll publish at a later time, maybe episode 3 or 4. It's work in progress. Long story short, the top row, you see how this is done with the patients going in. Middle row is then the huge amount of data analysis and literally data cleaning that you have to do. And the bottom row is then finally you end up with an analysis. In the cases of EEG, for example, EEG is very, very sensitive. So you can think you have the person inside an fMRI scanner tube, like we saw in the previous one. At the same time, they have a cap on, and at the same time, they actually have to keep the head in the same position. So they're reasonably, not tied down, but they shouldn't move too much. They shouldn't open their eyes and close their eyes too much, because all these things get directly into your acquisition system. If you open and close your eyes, you get a signal on the EEG. If you move your head, you get an artifact in the fMRI. So in order to get rid of all this, purely technical data collection, cleanup process, that's what you do in the middle row. And then you finally end up with some the data at the very bottom. In the case of fMRI, you get imaging data, color pictures, or you get quantitative data like bars, for example. Yeah, like you saw in that slide. So that is and that, you need to be, I've tried to educate myself, and we all at GABA to have in EEG and fMRI data analysis. But at the end of the day, we did this together with Mindig, and these guys are really top guns, in my opinion, regarding EEG. They are actually, pioneering and driving very much forward both a quantitative and high density EEG, because you can think the more electrodes you can put onto one skull, the more data you can acquire. And it's also good for us because we had the same guys analyzing both the fMRI and the EEG data, because it's the same data package looked at once you extract the fMRI data or the EEG data previously, but it's done by the same guys. So they access our same data, our same data room that we had. So that's very good, I think, from a general, say, data analysis point of view.
I think also something that's quite good with the study that you have conducted is that we see that you have visit 1, 2, and 3, and these are randomized, right? With the placebo,
Yeah.
you have alternative drug candidate, and you have GT002. Could
And
you?
it's exactly so. It's randomized. So the people, you have to, at the end of the experiment, once it's actually finished, then you can of course open animal, and then you can put them into different groups. But in the moment the people are analyzing it, they don't know that yet. So it's the same design. But again, remember, this is healthy volunteers. So things like if you say, oh yeah, but we didn't see so much dramatic effects. I don't think that you should go and expect and hope for very dramatic effect in healthy volunteers. These are young people. They're actually quite fit. They're actually, for example, if you think about the memory task, okay, so it's very difficult to improve the memory in people that are reasonably young and fit and university students, most of them anyway, as compared to a group of, say, my age group, is old people that forget stuff. So that's a sense that in the participant group, the main thing was to make the baseline now. And now we can see what happens in the EEG and the healthy volunteers. And of course, this information will go directly, for example, as we go on later on, what areas of the brain are affected. Okay, now we know what to look double or triple or 4 times as closely as possible in the patients, So it's a little bit of, if you're looking for a needle in a haystack, it's a good thing, a good idea to know, okay, this part, the lower right-hand corner of the haystack, there, let's look 2 or 3 times more closely than we would do compared to all the other rest. I think that was the basis of the idea that we had behind this fMRI EEG study as a bridge to the patients, So most important for me in the fMRI data that we got and we did the press release about was, that the data had to be convergent. Convergent just is a fancy word for saying they all go towards the same goal. They point at the same result to get to the same data, so that the networks that we're activating make sense, that what you see in the EEG also makes sense that what you see in the fMRI. And that's what I've been doing, since we got the data in. We got the data. I worked a lot with AI. I worked a lot with the literature that I have in the library. We have circulated this information to the people that are, opinion leaders in the field. We get the feedback from them next week to see what they think. What I what myself and AI think, at least, that is consistent, that the oscillations that we see in the EEG and the low frequency connectivity that we see fMRI, they fit together. So it's a validation of we had said what we want GT002 to do. We also have shown that the effect is specific, because in both an fMRI and EEG, since this has come from the same experiment, we have lorazepam as a control substance, same receptor, different chemical structure, reference compound, and placebo, which is just a sugar pill. So the effect of GT002 were selective. They were consistent with what we have seen in the EEG. We seem to be affecting mainly the low frequency range in the alpha waves in the EEG, the low frequency oscillations in the fMRI. And these 2 things actually hang together. So as a therapeutic potential, I think it's very interesting. In particular, I think I have in the next slide which areas, on this slide, which areas on the brain we're working with in fMRI. If you look at the next slide, if you look at areas that are relevant to cognitive and sensory motor, i.e., how do we process the information from our senses? This can be sound, eyes, touch, smell in the brain. And these areas that are, remember, that are enhanced, they are positively affected by GT002 as seen in the fMRI. And that is, of course, very, very nice. So you remember that in fMRI, what you're measuring is actually an activation of the brain blood flow in that particular brain area. You're measuring a signal that comes from the blood. And we infer then, okay, the more of this you have, bold signal you have, the more active that area in the brain is. In the EEG, you're measuring the signal the electrical signal directly, a little bit different. Long story short, these two are congruent. We have slow alpha oscillations, low frequency, we measured with the EEG, and we have fMRI, low frequency oscillations too. Both of them show that the areas of the brain, be it the parietal region, the frontotemporal region, in the case of the EEG, seem to converge that are relevant for things like cognitive processing, sensory motor gating, language functions, social interaction. So these are all areas that are, of course, extremely relevant to schizophrenia, and we think of pathology. They are relevant to those areas that we were interested in from the beginning. Because remember, if you go 20 steps back and you look at my presentations from the last many years, I've always said the same thing. What I want to show in this study is we give GT002 orally, we affect the right areas in the brain. The effect is different from the drugs that are already on the market, and it's safety and well tolerated. And that we've proven with these two studies very, very nicely, in my opinion. And icing on the cake is that it's a nice bridge to the totem study, which we're starting with, because now we know which areas of the brain we have seen in the healthy volunteers. Now we can really have put in under particular scrutiny, and we know where to look now. What can we expect, and what difference we can see in the patient with the EEG study and the totem study? So it's a little bit, a bridge. It's not a bridge study, but it shows us which direction to look at in the next results that will be coming in. And remember, this is still only the people that are in the resting state. And it will be super interesting to look at once we put these poor people through the tests while they were in the tube with the EEG and the fMRI.
Can I ask about those tasks then? Because you had the resting state, right? And then you have the other states. But what do they mean per se?
So the tasks are very, very simple cognitive tasks, Marcus. I'm not going to put you through one of these tasks because it's one of the tasks is called the NVAC test, which is a very simple task. What you do is you say a sequence of numbers or letters or objects, and all then you have to do, okay, which was the third last one that I mentioned to you? If I mention you 3 objects or 3 numbers or 3 letters, it's very simple. You can make it very complicated, because nobody can remember the 5th letter I just told you, and so on. That's one. That's the cognitive test. And the other test where you do is where you look a little bit at the emotion. And what you then do is, you have people have to rate themselves, how they're feeling when they're exposed to certain images, for example. So these 2 data sets are still coming in, but I'm
very exciting, right?
I'm more than happy that we have a proper baseline for the next study.
Yes.
And in our next episode, I will make a slide, that this kind of project is actually, you have to stepping stones, that you have to hop over to the next, and to the next, and to the next stage. So what we have done now, we hop from imaging data. Okay, now we're next level. Now we can jump into the next set of data with the patients. Once we got that, then we can expand the number of the patients, because now we know where to look at. So this data was actually very nice, because it confirmed what we've seen with the EEG, and it allows us to look forward to the next result.
Let's do this then. We're very much looking forward for the cognitive and emotional tasks as well. But could we, if I can ask this question, when you say that there's a weaker transient effect for lorazepam or a placebo, what does that mean per se for a layman if they would look into Gabatter?
For a layman, that means, for example, if you have you start all your patients, all your volunteers at time 0. You give them a placebo. Then you start measuring the effect in the fMRI scanner, say after 2 hours and after 4 hours. Then you would see with GT002, you would see a very strong rise in the low frequency oscillation in a certain area of the brain. And if you do the same with a placebo, then you would see a very slow effect or nothing that disappears. Nothing really happened. And with lorazepam, then you would see probably a little bit lower like that. So you see the difference in the timing, and you see the difference between GT002 and the 3 different modalities. So a placebo effect would not stimulate those brain areas. GT002 would, and lorazepam would either not stimulate or much lower than GT002. So it's statistics significant difference between the increase in the signal strength that you're measuring.
Mm.
Which
I
is actually exactly
totally,
what you expect. If you give somebody placebo, you hopefully nothing happens. If you
yeah,
give
of
somebody
course.
a drug that you want to affect the brain, of course you expect something to happen, and in the right direction. Because what the last thing you want to do is that you suppress an activity in the case you actually you're looking forward that you have a pathology and what you want to improve the activity in that brain area. The last thing that you want to do, and you don't know, it's an experiment, that you
Yeah.
depress it. And this has not happened with GT002 at all. So that's good.
Because something that I was thinking of, maybe now I'm going back to the emotional tasks again, but side effects in this, would that maybe show itself in the emotional one rather than the other steps?
It could be. It could be.
Yeah.
I don't know. We've been at the site. We've seen the people in this tube, and nobody reported. It's a clinical trial, so nobody reported any side effects. So I don't know what the people actually scored once they said, "Oh, I felt stressed, tense." There are 6 different parameters that you actually get through, but we'd see that. Would that then also translate? Now you're really into experimental sciences, because then you have to say, okay, how would that score then translate into what kind of signal, either in
Yeah,
EEG or
yeah, yeah.
fMRI? But that is still too far. But let's see when we got the data. Definitively,
Yeah, of
I
course.
think.
But it would be interesting to see, right? Because we know that lorazepam is not the greatest when it comes to side effects as well.
Yeah,
So, and that's kind of the unique
uh,
selling point of
I,
GT002, right?
absolutely, absolutely. And so, in that sense, even that there is a consistency. If GT002, if we start from preclinical effect, and this is a partial allosteric modulator, and lorazepam is a full agonist modulator, then you definitely would expect lower side effect profile, or more mild profile side effect profile, and more
Okay.
specific one,
Yeah,
hopefully.
cool. Got sidetracked, but it's so interesting. So let's move on then. So
No worries.
I
Sorry.
think let's
Yeah,
talk about the status as of now.
apology.
Yeah.
I do get carried. So this is a little bit of
Oh,
a
me too.
repetition to put everybody to sleep at the end of this thing. The key message that all I wanted to say is, now, as of today, end of April 2025, we have an orally available, safe and well-tolerated drug. We have done 2 clinical trials to test single and multiple ascending dose. We've done an EEG fMRI trial. We looked at CNS effects. We have proven that the effect is there. We have proven that they're different from placebo and a controlled substance. We have proven that they're sustained, and they're interesting areas of the brain that are affected in EEG and MRI. These areas are, slide number 3, they are relevant to neuropathology. If you go back in the slides and you take a look, the areas of the brain mentioned in slide number 3 or 4, they're definitively relevant for all diseases where you look at cognitive deficits, when you look to social interaction. There you go. Parietal region, for example, is relevant for language and understanding. So these regions of the brain, they are relevant for the neuropathology. It's not the middle. Now, of course, you have to be careful. Once you talk about this kind of higher brain function, everything is connected with everything. So it's not reasonable to expect that, if affect one brain area, you will have, 100% sure a very specific effect on that, on one behavior that you're looking at. But the areas are definitely interesting. It's consistent what you see in EEG and fMRI.
Mm.
And it's different to lorazepam. And that, I'm more than I've always, again, I remember these were healthy volunteers we looked at. They are the stepping stone for looking into the patient. At the end of the day, we're trying to develop a drug to hopefully treat some symptoms in patients, not in healthy volunteers. And that's what the last slide, the last little box here, cartoon on the right hand side, that they're consistent with each other. The EEG and fMRI, they fit together. So for a leading drug candidate, I think these 4, how would you say, characteristics, or these 4 features are actually very interesting. At least they would be for me.
Yeah. It's been a very long journey, for Gabatter so far. And it's cool to see that it's actually going for the preclinical now to actually seeing these results. Was it better than you hoped for? What's your feelings? Because there's a person behind this also as well.
Definitively. I mean, it's been, you know, a terribly long journey. Lots and lots of bottlenecks, I must say. But this kind of data is exactly what I had hoped for regarding this particular trial. You know, it gives us a platform. And for example, somebody comes, some clinician says, listen, I believe that this kind of profile could be interesting for my indication. I work with, pick whatever neuropsychiatric or neurology. I think that, you know, deficits in speech and language recognition or in face recognition is relevant for autism. Do you think that GT002 could help them? Well, let's take a look at the data, you know. It's right there. And if you think, and that's actually what we're doing now, we're going to the to opinion leaders, say, okay, this is our data. What do you guys think? And then there might be, you know, a new the project you can start right there. We've put the basis for the next studies is there. This is healthy volunteers. From then now you can extrapolate, okay, could this be interesting for a group of patients or for a certain group of symptoms? And I think definitively if I go, you know, speak to clinicians and said, listen, we have this clinical candidate drug. We are thinking about, do you think this could be interesting running a clinical trial in dementia, for example? We look at cognitive deficits. The first thing will happen, oh, is this relevant for dementia, Alzheimer's, and so on. So I think it's
interesting.
it's a
You have
nice
really
story.
swapped the order of everything, right? Like if you look at drug discovery, usually you look at some symptoms or some disease, and then you work your way towards there. But Gabatter has done it completely the opposite, right? You start looking at the receptor, and then you go
Exactly.
from there. So the possibilities are so broad.
That is perfect. Exactly what I should have said that, Marcus, but you took it away from me. Exactly right. Because when you, and that's why we need an episode on the history of GABA, because the
Yeah, we
GABA
should have
started at
it.
the very beginning. What we're going to do is we're going to synthesize a molecule, and then we're going to test them in what different models. And once we have the model, then I said, okay, this might be useful for this, you know, indication, and then we go that way. And that way, and as I've said, you know, we never started, like you said at the very beginning, said, okay, we have this symptom, so we will now make a company that treats, I don't know, depression, for example. We made a company that started with a bunch of molecules and 2 pattern groups, and let's see if we can make, you know, an interesting drug candidate out of it. And that we have succeeded. And then to come back to what you mentioned very nicely, Marcus, is it's been a long journey. I am really happy that we've got so far. Took much longer than I expected, but you know, once you're there, it's brilliant. And to be honest, in the moment that the clinical trial material will start, and I get the news from the Totem study, hey, we've given a GT002, or they don't know, we've given the first treatment to the first patient, I will really, well, I don't understand George
But there
W.
is some
Bush
excitement there, right?
with mission accomplished, but it will really, a big weight will be off my shoulders, definitively. And to make it even nicer with these results, you know, you can go with these results to anybody that is interested in running a clinical trial and might have an idea this could be a mechanism, or this kind of profile in fMRI or EEG could be interesting for my indication, definitively.
So we look at,
Yeah.
I know that we mentioned that you're still waiting for the data for the cognitive and emotional tasks,
Yeah,
but
yeah,
there's some other stuff as well. Uh,
absolutely. So, you know, if you look at what are the potential next steps, we can you know, investigate in the sense of more patients, links to task performance. That is what the data that is coming. And of course,
mm.
next thing you can do then model the pharmacokinetics, where you then look at what metabolites and what concentration of GT002 you have during the trial. We've done it in the first 2 ones, so we know that it penetrates the brain. And finally, you know, then you can do next step to that is then look at the connectivity inside the different areas of the brain, and then you become, you know, then you're doing more into experimental neurosciences. Actually, we're looking at, okay, how does, for example, the low frequency stimulation that we see in the fMRI, low frequency oscillations, does it actually affect also high frequency formation, and so on and on. So there's a whole science bit you can into. But for us, the key thing here is now we have actually narrowed down what can we expect, what should we look, and with particular closeness in the patient studies, and what spectrum in the EEG. And I think if you think about how much data cleaning you had to do it to get this data, then once you get the next set of data, then you said, oh, okay, brilliant. So let's in the first then we should look at the alpha frequency in this EEG study in the patients, because we know that we had an effect in the healthy volunteers.
Yeah.
Or if you do an MRI study, then you're like, okay, then we will take an extra look at the parietal region. Let's see. And then, then there will be this data then, talking about consistency. EEG and fMRI have to be congruent towards the same goal. And of course, the ideal situation is that the healthy volunteer data and the data that you get out in the patients eventually also have to be congruent. We saw an increase in, for example, the activity in, for example, the parietal region in a healthy volunteer. Okay. Now we take a patient who has a decreased activity in that particular, in the same region. We give it GT002, and we see a normalization. And then finally, it's really mission accomplished, because this is what is called curing a symptom from a scientific, from an imaging point of view. So, but now I'm getting a little bit carried away, because now we're talking about the future. But this is this would be an ideal future scenario, I think.
Cool. We're very much looking forward for the cognitive and emotional data as well. And that, I think that would be the dot over the I, right? And then we also wait for the totem study that's to be started. Is there anything you like? What's the status on that? That's one of the questions that I've gotten. So let's go over to the Q&A a bit. So one of the questions I've had, what's the status of the totem study?
Yeah, but the status is that the production of the material is going on. The labeling and the packaging should be next week actually, and once it goes from there directly to the pharmacy in Denmark to start the study. So it doesn't go via Gabather at all, but it's taken a very long time with the production. But there has been an issue with the producer rather than with us.
But you have everything kind of settled when it comes to all the permits and all the. It's just the practicality of getting the candidate to Denmark as of now, right?
Yeah, the material is there. It's a question of putting that in the proper capsule form. It's been sent from the people that hold the material to the people that actually produce those capsules. And once they are produced, then they go to the site in Denmark, and they are administered, managed by the site in Denmark, and the logistics there, they are 100% in place. I probably will be there anyway a couple of times now we are starting. But again, the key thing is, and I really hope that as soon as possible in our next episode or whenever, be able to talk about, okay, now we started and everything seems to be looking good.
Yeah, I had also it was great. I got a lot of questions. So one of the questions, you had this new emission not that long ago, and the question was how many from the board joined in the new mission?
Well, that's a simple question, because I joined, and I am a board member, and I did join, absolutely.
Next question that I had was it was a while back that you communicated that you changed the certified advisor from Corpora to Västra Hamnen. What was the reason for that?
The reason for that was that we changed from Corpora. We had a financial advisor, Arto, for emissions in the autumn last year, and when we needed to do a new emission, Västra were very active, very helpful, and I think they're very convinced of the Gabather project as a way of since we're working so close together with them, anyway. It was basically the natural partner to continue as a certified advisor from Corpora to Westerham. And I was very happy with Corpora, but I'm also very happy with Westerham. And I was particularly taken in actually by their enthusiasm for the Gabather project in general. And I mean, I'm not making this up. I literally spoke with them this afternoon. They're still working with Gabather. They want to get updates and reports. So they're very active as a partner.
Yeah.
And I think it's a good choice, a good team there.
Yeah. I think also last time I had this question, and last time we said that there are some conventions and conferences and so on that where the GT002 will be communicated. But I had this question, and I think we should look at it again. And it's how will the data be spread or publicized for other partners or any relevant institutions get to know about the results?
Absolutely. There was a meeting in Chicago on innovation and schizophrenia, where the people in Denmark presented the TOTEMS trial with GT002. So that was the first presentation, nothing to do with this data. But this data I'm going to publish it together with Mindig. We're going to do one publication with the EEG and fMRI data, basically, the nitty-gritty of the data that I've just shown, very general results, of course, with figures and plots and so on, in a scientific publication. I don't know what publication yet. I'm discussing with Mahmoud Hassan from Mindig where to publish that, and then that data we will then use as a posters to go to present scientific and of course also business development. And also, of course, we will also add then input from the opinion leaders that are looking at our data now. We need to get the feedback. I mean, the data is really quite new. And I will try to get the publication, it's in our interest and also in Mindig's interest to get the publication ASAP, of course,
Yeah. That's kind of leading into the second question. I think that during 2024, you said that you're having discussions with potential partners. Are there any more potential partners and ones in Denmark right now of the TOTEM study, or is there more people interested in the data?
Absolutely. I mean, there are more people interested in the data from both in Europe and the US. But it is always a question of it's a little bit like you, Marcus. What these guys are going to say. Oh yeah, that's really nice. Thank you for this fantastic data. But what about the data about the cognitive and the emotional tasks? Said, okay, guys. So, but this data is sufficient, I think, for generating definitively more, and this will be good data to go out with people interested in licensing now in May. We have another one in June. It's not only me; it's also board members that are active in getting the word out. The scientific publication, that will be mainly me driving it forward. And regarding partners for clinical trials, we have people that are interested in running clinical trial with Gabather, actually. It's a group
That's interesting, right? Because
in Stockholm, in particular, looking at dementia. We talked about this frontotemporal dementia, and with these results regarding effect in brain areas relevant for cognition, I understand the dementia studies are even more interesting. So we have we can submit this kind of data, for example, to a granting organization. You stand very strongly because now, okay, you have a clinical candidate and you have shown effect in the imaging, and you have shown that the effect is relevant in the brain areas relevant for an indication, be it Alzheimer or whatever. That really, peps up your chances for getting grants. And I mean, working already now. Middle of May, we have a grant together with Mindig on looking at EEG data in the patient study from purely scientific point of view. So we can submit this data as a clinical partner, as a biotech partner. I think it really increases your chances for getting those competitive grants that been looking for a long time.
Of course. I think the last question that I had that I got through the channels, through email and Discord, if you want to reach out to me there as well, was who's working right now in Gabother? Because you had mentioned earlier that you have tried to save as much money as you can, so you extend the runway.
Well, yeah, right now there's Christine Ryan and myself, the CEO at Gabat, working on operations. We got, of course, CFO who's working as on consultancy basis, and the board, you know. So it's in my opinion, it's as lean as it gets. I mean, this is we don't do experiments with this kind of boxes here behind me anymore.
Yeah.
Unfortunately, I literally don't have the time for that. But I'm very active with all our partners. Say Minnick, for example. I mean, I can promise you, I had more than one meeting about this data and what it means, because the amount of data is just huge, and the implication of it is also big.
That
So
has always
for
been
the
your
activity,
strategy, right? You
yeah.
try
So
to
for the
be lean and
activity,
then, yeah.
yeah. So for the activity that we want to do, you know, we have the lowest, the leanest management profile that you can do, because what we want to do is we want to get a partner and want to do another clinical trial. Both of these things I don't have to do with these machines, but you have to have people that drive it. This kinds of talks with you, for example, you
Yeah.
need somebody to do it,
Yes.
right?
Yes, then maybe we can try to end it at this point in time. And I think me and you, Robin, we have been discussing a bit what shall be the next episode. We're hoping that the emotional and cognitive results would be in, and we
Yeah,
have a guest that we could see maybe who would like to join.
yeah,
We have a
absolutely.
secondary guest as well that we could see, and then we also have this topic of the history of Gabadar, because it would be nice to, going back to how it all started and how we have arrived where you're at, and the amount of work you have put in these years, and then comparing maybe to what big pharma has struggled with over a very long time, where you, as a very small company, has actually achieved this.
Yeah,
Quite
you know,
remarkable.
absolutely, Marcus. I mean, you know, it is. Well, we have. I've already asked for the two guests. I think it will be absolutely brilliant. So people not always have to listen what I have to say.
Yeah.
They already by now they know exactly what I'm going to say. That's number one. Number two, I'm very much interested in, actually, because I think that the people that listen to this channel are the people that have been with Gabather for a long time and actually extremely loyal. You know, and I actually want to tell this story because the Gabather story is actually an interesting one. So if you go back by now many years, but it's a good story to tell, and I really love to tell that. So that is for episodes 3 or 4, Marcus, and
We stay
you know,
tuned for early June, I think we're planning. So initially we said 2 to 3 weeks. We'll try every month, maybe now, because we have seen there's some planning to do.
I think so. Yeah, I mean, and you know, b- I mean, for all everybody that's listening has listened to this for so long. You know, all credit to Marcus. It's really who is driving this, who've done this, and the feedback, at least we've gotten. You've done a great job, Marcus. So think about
I think
your
you're
next
part of this
your
as well,
career as a
so you don't give
webcaster
me
is
all
on.
credit. No, no, but remember then, you have our contact information. If you have questions, you can send them my way. And then if you just have opinions on what was good, what was bad, what could we do better, just reach out. I think if you come with feedback, we can make it better, tailored for the ones listening.
Yeah,
Shall we say it like
absolutely,
that?
absolutely.
Thanks for
Thanks a
today,
lot,
Robin.
Marcus, and looking forward to episode 3.
Yes. Cheers, mate.
Okay,
Bye.
cheers.